Hello and welcome students once again to this ICSE biology series. This is Mrs. Ranjana Dubey who is taking you forward in this ICSE series जहाँ पे हमने chapter cell के बारे में पूरी तरह से discuss कर लिया है and after that I just started with cell division, and before I started with cell division I actually gave you all the important an explanation into all the important terminologies that we would be requiring to take the lesson cell division forward, and also the chapters like genetics, etc. So the things that we have discussed so that I can take up the chapter cell division we did discuss about what a chromosome is, what a chromatin fiber is, and what is a centromere, and chromatids, etc. So all these familiar sounding terms have been explained in my previous lectures. So you can have a look at those lectures when you actually come to see this topic on cell division. So this is the second topic on cell division. In the previous class of cell division, we have seen what cell division is and what is the need for a cell division in multicellular organisms. Like for example, now we know that all the organisms, the human beings and other organisms, they are made up of trillions and trillions of cells. But actually those cells had their origin as a single celled zygote. So this zygote multiplied and you have this trillions of cells. Trillions of cells getting formed, and this process of division, cell division, that helped in this was mitosis. This is the actual name of cell division, and we have another division which is we know by the name of meiosis, and I just try to explain the differences between the two terms by doing a comparative analysis between the two. We also said why there is a need for the cells to divide. Why can't we all live as single-celled organisms? So the answer to this was that we can ensure better functioning, better adaptability, more evolved organisms. When the number of cells are more, you can achieve a lot of division of labor, and you see organization of like cells. They will get organized to form tissue, which will organize in turn to form specialized organs, and then organ systems, like you have your digestive system, the endocrine system, nervous system, and so on, and eventually a multicellular organism. Okay, now we also said why do cells divide? So why do cells divide is mainly we did answer by saying that cells divided for the purpose of growth and to achieve development in the organisms, in the higher organisms. It was a means of replacement of the old, worn out, the dead cells with the new cells. Then the injured cells will be replaced by the process of cell division. So that is for the repair of the cells, you require the cells to divide, that is the cells to undergo mitosis. And for the purpose of reproduction also the cells divide, and which we see usually as in lower organisms as a form of asexual reproduction, like in amoeba, which divides into two, then bacteria which divide into two. So it is also going to serve as a process of reproduction in case of lower organisms, the process called cell division. And then what did we say? We said that the cell division is actually achieved through a cell cycle, which is nothing, what is it? It is nothing but a series of events or set of events from the birth of the cell, that is from the time the cell has been born. Like suppose if this is a cell, if this is the nucleus of the cell and this is the cytoplasm of the cell. So the time it divides into two cells. Now when it is divided into two small cells, obviously the newborn cells will be smaller in size. They will attain their full growth first. They will grow in size, and then they will achieve the reproductive ability, and eventually they will also divide into the two daughter cells. And I also said as to why am I always calling them daughter cells and not son cells. That is nothing because I am prejudiced or a hardcore feminist. It's because these daughter cells are also going to grow in size and eventually give rise to next set of their daughter cells to continue the progeny, to ensure that the species continuation is always achieved. So this sequence of events from the time when a cell is born to the time you get daughter cells is nothing but a cell cycle. And this cell cycle duration, it varies from individual to individual, cell to cell. It is not the same in all organisms. Now the cell cycle is in turn divided into two broad phases, as you see here, the interphase and the mitotic phase. So what is this M phase or the mitotic phase? I'll write here. Meiosis also, like these are the two divisions that we talk about, mitosis and meiosis. So this is the actual phase when the cell is dividing. So this is the dividing phase, that is when the cell is actually splitting into two cells or four cells if it is a meiosis division. So this is the dividing phase, and before the cell has divided, it will rest or it will remain in the interphase. Okay, then take a look at this diagram. This interphase, children, is further divided into, I said it is further divided. Now let me take the black color. This is in turn divided into G1, S and G2 phases. Yes, so this is what we talked about in our previous classes. I said that the M phase or mitotic phase, as you can see in this particular diagram, which is further divided into four phases, which is prophase, metaphase, anaphase, and telophase, which we can abbreviate as PMAT. PMAT, that is prophase, metaphase, anaphase, and telophase is what the M of the mitotic phase is in turn divided into, and this is actually also called as the Karyokinesis, that is the division of the nucleus. Now, after the division of the nucleus has been achieved, for the cells to actually function as two cells, it has to have a certain amount of cytoplasm and some other cell organelles also. So another set of division is called as cytokinesis also takes place, which is nothing but the division of the cytoplasm, the formation of the various cell organelles, and then their incorporation of these cell organelles into the two daughter cells, so that when the daughter cells are actually formed, they don't just have a functional nucleus with them with the genetic material. They also have certain amount of cytoplasm and other cell organelles which are also needed for the functioning of the organism or the cell as an individual cell. So for the cell to function, it not just requires nucleus, but it also requires cytoplasm so that the other metabolic activities also can go on, and it also requires various cell organelles, like for example plastids. If it is a plant cells, it will require lysosomes, it will require vacuoles, it will require mitochondria, it will require various enzymes and proteins to carry on the various metabolic reactions, lipids, formation of various lipids and other chemical compounds which would be required for a cell to exist as an individual cell. So this is what we had also done. We said that the M phase, or the actual phase in which the cell is dividing, it is divided into number one, which is karyokinesis, which is the time when the nucleus of the cell divides, and this is achieved by PMAT, that is prophase, metaphase, anaphase, and telophase. Then after the nucleus of the cell has been divided, as you can see in this particular diagram, it is the cytoplasm. We need to divide a certain amount of cytoplasm into the newly formed two nuclei and also give the cytoplasm certain cell organelles so that the cell can start functioning as an independent cell. So the next division is the cytokinesis, which is the division of the cytoplasm, and only then you have two functional daughter cells coming into existence. So this is what we had covered in the previous class. So the topics to follow would be more detail into the process of cell cycle, wherein we will see or we will discuss more about largely the interphase. That is the phase in which the cell is spending most of its time, interphase. We have also discussed the cell duration. So while discussing cell duration, we did say that interphase is the phase of the cell cycle in which the cell spends most of its time, rather 90% or 95% of the time of the cell is actually spent in interphase. So we will be discussing in detail about the interphase. That is, we are going to discuss about the G1 phase, the S phase, and the G2 phases of the interphase. And then we finally go on to study the actual M phase or the mitotic phase, wherein we discuss the individual stages, pro, meta, ana, and telophase in detail, after which we will be taking up the significance of mitosis, that is, what are the largely things that we are going to achieve in a particular organism or a species by the process of mitosis, and finally the differences between the mitosis in the plants as well as animal cells. So this is what are the broad topics that we would be taking up in studying mitosis, the cell division. Now, in the previous class, we said that the cell cycle duration is variable from organism to organism. Okay, what is, you can have children look at this particular diagram. Now it is a representation of a cycle. That is, a circle has been drawn, and this circle is divided into various quadrants to depict the various stages that the cell undergoes or remains in before the division takes place, or actually when the cell is dividing. Children, what do you see here? This is the mitotic phase. So the phase in which the cell is dividing, mitotic phase is also the dividing phase. I can write down here as the dividing phase. So can you see what is the length? This is so small. The phase in which the cell is dividing, isn't it very small as compared to this particular phase or this particular phase or this particular phase? Yes, we can clearly make that out, that the karyokinesis, that is mitosis, is a very takes place for a very, very small duration. Okay, like suppose if I were to divide this circle into 24 hours, because 24 hours is the cell duration of a typical mammalian cell. So out of the 24 hours that a mammalian cell spends for completing its cell cycle, only 1 hour is spent in the M phase or the mitotic phase, and remaining 23 hours is spent in the interphase. That is, what is interphase? Interphase is nothing but the preparatory phase. It is nothing but the preparatory phase. Why do I call it a preparatory phase, children? I call it preparatory phase because this is the phase in which the cell is preparing to divide into two daughter cells. Okay, so this is also called as the preparatory phase. So that means to say that preparatory phase, which is lasting for 23 hours, so that means which we call as interphase, we have also called it as the resting phase. Now that does not mean that the cell is resting or sleeping and it is not doing any work. It is rather no major changes you are not able to look at in the resting phase, which is why or interphase, which is why we also call it as the resting phase. But there are a lot of activities that are taking place in order for the cell to undergo M phase, or to actually split into two cells or two daughter cells. Okay, now what are the various preparations that a cell would be requiring in order to split into two? Now this you can understand by, suppose, taking an example of a man who is also a father, and he has to give his entire property to his children. Suppose he has got two children, and he has to divide his property among his two children. So what will he do? First of all, he will acquire that property. So suppose he has got few houses, land, cash, gold. Yes. So all these tangible assets, they need to be first created. Yes. And then they need to be divided equally between the two offsprings or the two children that the man has. So suppose he has got two houses, and he would be giving his one house to one son and another house to the other son. Suppose it is a land, so if the land also will be shared equally between the two. So that means what, each and every part of the property, whether it is land or whether it is gold or whether it is cash or whether it is in any form of property, whether it is a house, it has to be equally divided among his children. Yes. So that means he should be having enough of the things so that he can divide, or whatever he has, that has to be divided equally among the two offsprings that he has. Now, in case of mitotic division, what have we seen is that the daughter cells are genetically completely identical to the parent cell. So that means what? That the genetic information or the DNA, which are the carriers of the genetic information, they need to be transferred to the daughter cells in the same quantity as they are present in the parent cell. Yes, the DNA or the genetic information need to transferred to the daughter cells such that they are genetically identical to parent cell. Yes. So what will the cell do is that it will acquire everything that it needs. It will double those things, and then it will divide itself into two, so that one set of things are transferred to one daughter cell and another set of things get transferred to the other set of things. And the entire preparation for this actual splitting and the division of all the contents of the cell in equally to the two daughter cells, that preparation is achieved in the interphase through the G1S and the G2 phases, which are the parts of the interphases. Okay, so let me just read through this slide. Growth occurs during the G1S and G2 phases. Now, why growth occurs? I told you that the G1 phase, or the first part of the interphase, it begins just after the cell is born. It is just after a cell is born. Like suppose here, these are the two daughter cells which have been formed from this parent cell. Okay. Now this daughter cell, they have been formed at this stage. Okay. Now when they have been formed at this stage, now they need to grow in size, so that they need to grow in size. Only then will they be entering into another splitting, that is splitting further into the G phase. So they will grow in size, they will double all the contents that they have inside them, and only then will they be ready to further split into the next two daughter cells to continue the species. So M phase, children, lasts for an hour in human cells, and the remaining 23 hours are spent in the G1, S and the G2 phases. We see that the most variation of length is seen in the G1 phase. The G1 phase, since the cell is growing in size, that it is acquiring, and it is acquiring the things or amassing things inside itself so that it develops the reproductive capacity or ability. Yes, so the time which is spent and which we call it as the G1 phase, it is variable in the different cells. It is not constant in all types of cells. Okay, now children, the major things which you see are in the M phase when the cell is actually splitting, the M phase when the cell is actually splitting, and then the next major thing that you see is in the DNA, that is the S phase, when the DNA is getting synthesized. Take a look at this circle, children. The X phase is the next important phase when the cell is, when you see major activity taking place, that is the DNA or the synthesis of the DNA taking place. Yes, and M phase is the phase when the cell is dividing. Now, in between the S phase and the M phase, the cell spends some time, which is also called as the gap 1 phase or the growth phase, where the cell is increasing in size and amassing things that it needs for the cell to enter the S phase, where it can do the DNA synthesis. Okay, now once the cell has done DNA synthesis and it is almost ready to divide into two, it once again spends some time or takes some rest or goes into next part of the interphase. So this second gap period, when the cell has divided its DNA or multiplied its DNA, but is not divided completely into two, the cell is not divided completely into two. So that second gap period wherein the cell is present is called as the G2 phase. What happens in this G2 phase, we will be seeing. Now next, what happens is that, children, now just as when the cell has divided, after entering into the G1 phase, where the cell has started to grow in size, it says that I do not want a, it quits. It quits the cell cycle and it says that I will not enter the M phase or the S phase or the G2 phase because I don't have to divide into two. So some cells which do not divide, which after the G1 phase, they do not enter into the S phase or the G2 phase or the M phase where they are dividing into two. That stage is called as the G0 stage, and we have said that some cells, like for example our nerve cells, some muscle cells of the heart, even the RBC cells, they also do not undergo the M phase or the S phase because they decide not to split into two or divide into the two daughter cells. So children, please be careful. The G0 phase, where is it achieved? The G0 phase is achieved after G1 or gap 1 phase. Remember, this is important. At which stage do you observe the cells entering into, or some of the cells entering into the G1 phase? It is the G0 phase. It is the G1 phase. The cell has completed the G1 phase. It has grown in size, and now it decides that I am not going to prepare myself for further division into the two daughter cells. What I will do is that it will quit itself. It's called as the quiescent stage, where it simply does its work. It is a live cell. It is continuously performing its function after it has attained its full size, full capacity, but it does not divide into two. So therefore, it does not enter into the next phases like the S phase or the G2 phase and eventually the M phases. So it is important to remember that G0 comes after the G1 phase. Yes. So this is just a recapitulation of, children, what we have done so far. Interphase is also called as the resting phase. It is a phase between two successive M phases. Have we discussed this? Yes, we have discussed this. And what is achieved in this interphase, that the cell is growing in size, yes, so that it obtains or it develops the reproductive capacity, it develops the organelles in itself, it is able to multiply those organelles so that it is able to equally divide the organelles into the daughter cells. Then it is also going to do a lot of DNA synthesis in it. so that it is able to transfer the entire genetic material of the parent cell into two daughter cells. So that means what it needs to double its genetic information also, so that it is able to give the genetic information equally as it had in the parent cell. And we have already seen that this interphase is actually 95% of the entire cell duration. Yes, children? And this interphase, what in the interphase, what is the type of the genetic material, or what is the nature of the genetic material? The nature of the genetic material in the interphase, it is once again, it is in the form of chromatin thread. Yes, chromatin thread, which is what? Which is made up of DNA and some histone proteins. All this we have already discussed, children, in my preparatory class to take up the chapter cell division and genetics. We have already discussed the chromatin threads and what our chromatin fiber is actually, chromatin network is actually made up of. We said that it is made up of DNA, a nucleic acid, and some histone proteins along which the DNA is bound, because we said that the length of the DNA was how much? It was around 2.2 meters, and that has to be packed into an organelle called cell, and within that organelle, inside the nucleus of the cell, which has dimensions as low as micrometers. Yes, so that packaging is achieved with the help of certain proteins, which we called as the histone proteins. So in the interphase, we see we do not see chromosomes at all. What we see as the genetic material are the chromatin fibers or the chromatin threads made up of DNA and the histone, which are all simply, as you can see here, in this format. Yes, so in order to achieve, sorry, the splitting of this chromatin fiber equally into two parts, it needs to undergo a lot of coiling and recoiling. Yes, and it is only when a lot of coiling and recoiling is achieved, that is when the cell completes interphase, that you actually get this shape of the chromatin fiber into this X-shaped. Sorry, children. Into this X-shaped form. What is this X-shaped form? This is an intense coiled form, where the DNA has itself coiled itself extensively. The chromatin material has undergone coiling and supercoiling and has achieved the X stage. Now, is it easy for the chromatin material to be divided into two? Yes, now I can divide it into two. Like I can do a splitting here and it will be divided into two equal parts. It's something like a bowl of noodles, a huge bowl of noodles. If I were to, they are all spread around like this, and if I ask you to divide them into two, will it be possible? No, it won't be possible. So what will I do is that probably I'll make a sweater of like the ball of wool. You either make a sweater of it or you roll it, and then you cut it into two. You can cut a piece of sweater into two with the means of scissors. Then you can divide it into two equal parts. But simply all the entangled threads of wool, if they are simply scattered like this, is it possible to equally divide it into two equal parts? No, it is not. So for that, I require to do its coiling and supercoiling. And when I do that, I achieve that X-shaped structure, which I call it as chromosomes. So can I see chromosome in the interphase? No, I cannot see chromosome in the interphase. So I will also write it down. Chromosomes are not visible in interphase. Then when are they visible? They are visible when the interphase has completed, the DNA has duplicated, and the genetic material or the chromatin network has supercoiled itself, and that happens in the M phase. That is the first part of the M phase, which is called as the prophase. Pro means the early or the beginning. So it's called as the prophase. And in the prophase, the supercoiling of the DNA has been achieved because it is getting ready to divide into two. So it is getting the shape in which it is able to be equally divided into two equal halves. It is easier for a cell to divide into two equal halves. So the shape that makes it or allows it to happen, that particular shape is the X-shaped, which is what is called as the chromosome, which is the supercoiled stage, which we are able to see only after the interphase has completed and the cell has eventually entered the M phase, and the first part of the M phase is called the prophase. So in the prophase, you are able to see chromosomes. Okay. Children, at this phase, this stage, I would also like to say that there are no, as such, phases when the cell is actually divided into two. Now, don't misunderstand by what I just said. It is for our simplicity and our understanding that we are giving various names to the phenomena of cell division, like we are splitting the phenomena of cell division by calling the words or by using the words interphase, then M, which is further split into prophase, metaphase, and anaphase, and telophase. It is for our understanding and simplicity that we have given the names prophase, anaphase, telophase. There is no such clear-cut demarcation of phases that is taking place. So our understanding and our simplicity, we have given these names so that we can actually eventually analyze and say that these are this list of events which took place in the interphase, and then these are the events which are taking place now in the M phase, which is prophase, metaphase, or anaphase, and telophase. Now coming to the first part of the interphase, children, which is called as the first gap phase, which is also called as the growth phase. That is, the cell has been born. This is the new cell. Now, what did children go back to the chapter cell and recapitulate the postulate of Rudolf Virchow? What did he say? He said that all cells come from the pre-existing cell. So this is the new cell that has been formed. So any cell comes from the pre-existing cell. Yes, then what happens is that this cell will, the newly formed cell will grow in size, children. Yes, so it is growing in size. So I call it as the growth phase or the G1 phase. It is the first resting phase wherein I only see the growth of the cell takes place, the growth in the metabolism, cytoplasm taking place. In order to achieve growth, what is required? What is required? Tell me. For us to grow in, what is required? We require a lot of energy. We require a lot of raw materials or food is required. Yes, only then can we grow in size. So for the cell also to grow in size, it also requires certain raw material, amount of energy, etc. So what is that raw material? That raw material is in the form of proteins, children. Proteins or enzymes. Now, what are enzymes? In the presence of enzymes, the reactions can take place, various metabolic reactions can take place, and the cell can achieve the growth in its size. Okay, so for the enzymes to function, they need to be created first. Yes, what are enzymes? They are nothing but proteins in nature. All enzymes are proteins in nature, though all proteins need not necessarily be enzymes. Okay, so remember this word always. All enzymes are proteins, but all proteins are not enzymes. That is, it is not necessary for every protein to function as an enzyme. What is an enzyme? It is nothing but a biological catalyst, isn't it? So in order for a protein to function as a biological catalyst, we use the term enzyme. And this biological catalyst will bring about the various metabolic reactions. Metabolic reactions. Okay, so to carry out the metabolic reactions, we require enzymes. And in order to form a lot of enzymes, we have to do the process of transcription and the process of translation. What is transcription and translation? Children, the DNA, the information to form proteins is only present in the DNA, which resides into the nucleus of the cell. So what happens is that the information from the DNA, it passes on to the RNA, and this RNA then helps it actually prepares the protein, which are the bearers of the information of the DNA, which will catalyze the further reactions. So I can write here enzymes. And this process of the formation of DNA to RNA is transcription, and from RNA, or the overall process of the formation of proteins, is called as translation. So in order for the cell to grow in size, you require enzymes and proteins, and in order for enzymes and proteins to be formed, you need to carry out the processes of transcription and translation. So these are the events that will have to take place if the cell has to grow in size. So that is, these are the events which will take place in the G1 phase. So let us give a reading. Main events are the cell is growing in size. So, and it is preparing a lot of proteins and enzymes and making the cell metabolically active, that it is carrying out a lot of reactions with the help of those created enzymes and proteins through translation and transcription processes. Yes. Then after the G1 phase, the cell has to get ready to divide into two. So what it needs to do is that it has to pass on the genetic information into the daughter cells. Yes. So whatever genetic information it has, now the cell does not have to, one cell does not have to form one cell. Suppose the genetic information is X. Suppose the genetic information is X. So the new cell formed will have the X amount of genetic information in it. But this will happen only when one cell is going to form. But if suppose two cells are to form, and this also is to have the same amount of genetic material. So the total genetic material becomes X plus X, which is equal to 2X. But the parent cell, but the parent cell child, this is the parent cell. If the parent cell has got only X genetic material, then obviously it cannot transfer X genetic material as 2X in its two daughter cells. So what it needs to do, it needs to double its genetic material from X to 2X, and only when it has doubled the genetic material from X to 2X, that it can pass off the genetic information as X and X into the two newly created daughter cells. So this doubling of the genetic material, is the next important series of event that has to take place after the cell has grown in size. Yes. So to double the genetic material, that means double the amount of DNA. What is DNA made up of? DNA is made up of nucleotides. Yes, chromatin fiber is made up of DNA, which is made up of nucleotides, and it is wound around histone proteins. Yes. So that means what? It needs to prepare a lot of nucleotides, and it needs to prepare a lot of histone proteins, double the amount of histone proteins also, because since the DNA would be doubled, so histone proteins also need to be doubled, so that the same quantity of the chromatin material or the genetic material is created. Yes, so all this need to be done in the for the cell to have or acquire or double its genetic material. So that means what? The cell will prepare everything that is required for it to do the doubling of its genetic material. It will prepare a lot of nucleotides, it will assemble a lot of nucleotides, it will double the quantity of the histone proteins, okay? And a lot of other proteins would be prepared, for which it requires a lot. For in order to prepare protein, children, RNA is required, and enzymes are to be required for the process of various chemical reactions. So therefore, which are enzymes are nothing but proteins. So that means what? A lot of RNA and proteins have to be synthesized. These are very, very important and remarkable events of the G1 phase, because the G1 phase will ensure that the cell grows in size. It will ensure that the cell has got the ability to double its genetic content. So in order to get that ability to double its genetic content, it should have the necessary raw materials. So what are those raw materials? Those raw materials are RNA, proteins, enzymes, and so on. So these major things would be synthesized or formed in the G1 or the gap 1 or the growth 1 phase. Okay, and we also see increase in the number of some of the cell organelles, because the cell organelles also need to be divided from one parent to two parent cells, two daughter cells, right? So they also need to be increased in number so that it can be divided equally into two daughter cells. That is, the same quantity from the parent needs to go to not one but two cells. So that means other organelles also need to be doubled, not just the DNA content. but the other contents on the cytoplasm and the organelles of the cytoplasm also need to double themselves so that they can be divided equally into the two daughter cells. So these are the major events, children, of the gap 1 phase. It is going to grow in size, children. The cell is highly metabolically active. That is, for growth, it requires to perform a lot of activities for which it requires a lot of proteins to be formed. That is, transcription, translation will go on, and for which you require a lot of RNA and proteins to be present. So all these would be prepared a lot in the G1 phase, and we will also see an increase in the number of cell organelles, though largely the number of cell organelles we see in this doubling of the cell organelles we largely see in the second growth phase, that is the G2 phase. Okay. Now coming to the next phase, which is the S phase. S stands for synthetic phase. What is getting synthesized in the S phase? It is the DNA is getting synthesized, or DNA is getting doubled. Okay, it is the longest. Oh, it's not the longest, I would say, children. This is a wrong statement. G1 was the longest phase. Okay, then the important event is the DNA replication. What is replication? It is the duplication. DNA duplication is taking place here. The amount of DNA per cell doubles. So suppose if what has been shown in this diagram, we have shown 2 chromosomes. Now we have shown you 2 chromosomes. Yes. Now suppose if these 2 chromosomes are present, and this is the content of the DNA in first chromosome, and this is the content of the DNA in the second chromosome. Yes. So let me mark them as first and second. So I have to double my chromosome. So can you see this diagram? The DNA has doubled itself. Can you see the doubling of the DNA taking place? Now the DNA content has doubled itself. So if the DNA content was C here, the DNA content here is 2C. Similarly, if the DNA content was C here, the DNA content is 2C. Yes? And we already know what sister chromatids are. Sister chromatids are the arms of the chromosomes which are held by the same centromere. Okay? And this doubling of the DNA, it ensures that only the content of the DNA is doubled. The chromosome number is not doubled. Now, can you see the number of centromeres? Is it changed? The number of centromeres is still the same in these 2 doubled DNAs. Yes, this is chromosome 1, this is chromosome 2. The number of centromere is still 1 as it was present here. So, the number of chromosomes we will count by the number of centromere. So when the number of centromere has not changed, that means the number of chromosome also has not changed. Okay. So this is important, children. You have to know that the chromosome number does not change. It is the content of the DNA which changes from half to doubling. Okay. Now in animals, children, another significant event takes place is that not just we see the doubling of the DNA taking place, we also see the doubling of the duplication of an important cell organelle which we call as the centrioles. Centrosome, which have a pair of centrioles inside them placed at right angles to each other like this. Okay, so we see the doubling of the centriole also taking place, or beginning of the doubling of the centrioles also starting in the S phase itself. Now some of the books give this as the G2 phase, wherein you actually see the duplication of centrioles or centrosome taking place, but so in some books you see it written under S phase. So don't worry about all these things. We will say that the duplication of the centrosome begins in the S phase along with the duplication of the DNA. Okay, and it completes or finishes in the G2 phase. Now, this is another diagram of the S phase. What is happening is that before replication, that is before the DNA has been doubled, yes, this is the centromere that has been labeled, children. Yes, what happens is that you can see that the DNA content has doubled itself. These are the sister chromatids and they are called as the homologous chromosomes. So the chromosomes which contain similar types of genes in them and at the same location, and they are almost of the same sizes, are called as the homologous chromosomes. One pair of the homologous chromosome is contributed by one parent and the other is contributed by the other parent. All that has been discussed in the previous class. You can just go and have a look about these things. Okay. Now coming to the G2 phase, which occurs after the DNA content has been doubled and after the centrosome also has been doubled, typically in an animal cell. We see the second growth phase. Okay, the cell is getting almost ready to split into two, for which what it will do is that it will further grow in size. It will amass more and more of cytoplasm and will prepare more and more of RNA and proteins because they are required. And we see largely the multiplication of cell organelles. Now, we need to divide mitochondria also equally into the two daughter cells. We need to divide the chloroplast, we need to divide the lysosomes, we need to divide the various proteins, RNA, cytoplasm, and all the other granules which are present equally into the daughter cells. Yes, so so those organelles also need to be divided into two. So that really completion of the duplication of all the other cell organelles or multiplication of all the other cell organelles is completed in the second gap phase of the G2 phase. We see more and more synthesis of RNA and proteins here, children, so that the cell is now actually ready to enter the M phase. Yes, what can you see here? The cell has grown to its full size. Yes, and the centrosome has duplicated. The centrosome has duplicated. The chromatin thin thread would begin to start condensing. Okay, you can see the beginning of the condensation of the chromatin fibers, and all the RNA and proteins and the other cell organelles will get duplicated in the second growth phase. So we have completed sorry, the entire interphase, children, wherein we have seen the cell undergoing the G1 phase where it has grown into full size. Then it will synthesize a lot of RNA and proteins, that is, do a lot of transcription and translation. And some cells which will decide not to divide, like the nerve cells, etc., they will enter into the G0 phase from the G1 phase. Those which have to divide will then undergo the S phase where the DNA content and the centrosome will get duplicated, and all the other cell organelles also, which need to be equally divided among the two daughter cells, they will also be duplicated in the second G phase, that is G2 phase. Okay, and after the G2 phase, we will enter the actual M phase or the mitotic phase. So before I describe you the details of the M phase, let us go through some of the important points that we talk about mitosis. Mitosis is seen in all the somatic cells. What are somatic? That is body cells. So any cell of the body from top to bottom, you will see what kind of division taking place in it. The mitotic division taking place in it, because still the organism is alive. Every cell needs to function, and for every cell to function, it will undergo wear and tear. It will get injured. It will have to be replaced by new cells. So the mitotic division will take place in all the body cells till the organism is alive. Since equal amount of DNA and it is exactly equal to the parent cell, the new daughter cells formed are exact copies of the parent cell. So therefore, this type of division is also called as the equational division, as the number of chromosomes in the progeny cells remain the same. And in which type of cells do you see mitosis? We see mitosis in the diploid cell. That is the cells which have got 2n number of chromosomes. That is, the chromosomes are paired. Yes, in human beings, we have got 23 pairs of chromosome, one pair coming from each parent. Yes, so every cell which is diploid in nature, now are our cells diploid or haploid? Well, all the body cells are diploid except the cells which have been formed through meiosis. Okay, the sperms and the ova, these are the two organelles or the two cells which are created through meiosis. So they do not contain diploid number of cells, rather they will contain haploid number of cells. There is n number of cells. Okay, but does it happen even in the haploid cells? Yes, we do see it happening even in the haploid cells in lower plants and animals. Okay, for the sake of which undergo division as a means of reproduction. Okay, we see the mitosis also taking place in the haploid cells of certain plants and social insects. Then this involves major reorganization of all cell components. Now children, why does the cell undergo mitosis? Why does the cell has to divide into two? Because there is a kind of an imbalance that has been created as a result of cell growth. As the cell grows in size, what happens is that there is a disbalance in the amount of cytoplasm and the nucleus, or rather I would say the surface area, the cell volume, they are not in harmony. So there is some kind of a disbalance or disorientation. So in order to stabilize itself, what happens is that it will divide into two. So this will cause a major reorganization of all the cell components. So you can really understand it from in the same way as you understand the process called as nuclear fusion or fission. Why do you see various isotopes? Why do you have three isotopes of hydrogen as protium, deuterium, and tritium? The three isotopes of hydrogen are there mainly because in the third case, what you see is that there is a difference in the number of protons and neutrons. So when the difference is large, when the difference is more, you see the element starting to behave as a radioactive element. It begins to release certain rays in order to stabilize itself, and it starts to split. It is not stable anymore. So the same happens. So when the contents of the cell are not in harmony, that is, they are not grow. When they have grown in size, there is a disbalance among with the volume and the surface area, with the cytoplasm and nucleoplasm. Then the cell decides to divide into two. So cell division or mitosis also causes a major reorganization of all the cell contents. So let us stop here, children. So among the various topics of cell division, I have tried to explain you the main events of the cell cycle, wherein I have covered the G1 phase, the S phase, the G2 phase, and we talked a little bit on the major events of the mitosis, which is also called as the equational division taking place in the somatic cells, causes a major reorganization in the contents of the cell. In the coming classes, I would be taking up the major events of the M phase, which is pro, meta, ana, and telo phases. So I hope you enjoyed the class, and do keep watching the videos for more and more of such classes, so that your revision of your ICSE for the ICSE preparation is also going on simultaneously. So thank you, children. Thank you very much. Hope you are liking it. And in case you have any doubts or any query, you can post it in the comment section. Thank you very much, children. Bye-bye and have a nice day.